But is there a pathway to making this more of a diagnostic test? Because I would think that would be something that would be useful. Perhaps.
Yes. That's a really good question, and I'm glad you asked it, because I'm asked this all the time. So, is there a diagnostic test? Or oftentimes I get sent sort of requests to, can you sequence my poop in your lab? And I'm like, well, number one, I can't, because that's not what my lab does. But number two, there's some real dangers associated with that. And one of them is that, what are people going to do with the information that they get? And so what I'm seeing is that there are some private labs popping up that will propose to sample your gut microbiome, and usually stool sample, and tell you what microbes you have, and then tell you a bunch of stuff based on the results that I can tell you as a scientist are absolutely based in no fact whatsoever. The methods that they're using are not very robust. They don't seem to normalize anything, and I think it's dangerous, honestly, because quite honestly, we don't know what a healthy gut looks like. We don't have a definition for a healthy gut right now. And that's one of the reasons for doing something like the American Gut Project. We need to sort of set the baseline for that. And because there is so much variation among a population, it's very, very difficult to draw comparisons. The other really dangerous thing, or not dangerous, but thing that people don't kind of appreciate, is that when you do a test like that, you're taking a test in a snapshot of time. And your microbiome, although it's very kind of homogeneous and it stays very similar through your life, it changes in its abundance profiles according to what you're eating. And that's one of the ways that it kind of
What does that mean
copes.
when you say abundance profiles?
So if you think about your microbiome as a sort of a group of, 200 to 300 bacterial species. At any one moment, the numbers of each species are going to be different. So you're never going to have a ratio of 1 to 1 to 1 to 1. In fact, actually, it's usually very much skewed that you only have like 3 or 4 taxa which are really dominant, and the others are quite minimal. And that changes according to what you eat. And the reason it changes is because each of those microbes has a different kind of arsenal of enzymes to break down the food that you eat. Obviously, we don't all eat exactly the same thing every day, or most of us don't. So, and that means that that your microbiome changes in abundance profiles every day. And but it doesn't change in composition as much. So in other words, the same species are there, just the amounts of them are different. And so when you do a diagnostic test, usually when people do a diagnostic test, they're looking for a taxon, and they're thinking very much in terms of infectious disease, for example. They're looking for something that's a quote-unquote bad microbe, and if they see that in high numbers, then that's a bad thing. And the problem is that that's just frankly untrue, because it all depends on the context. And so you're not getting context at all if you're taking one sample. And even if you're taking multiple samples, I'd argue that the interpretation of the results that you get is just, it's not good enough right now. We just don't have the knowledge behind it. And that's actually one of the reasons why we don't, Health Canada doesn't have a test right now that they've sanctioned or for testing, because there isn't, nothing like that exists. So I would sort of say to people who want to do that kind of thing to save your money, there are lots of tests for pathogens out there which have been sanctioned and very clearly shown to be effective, and doctors have access to those all the time. But looking at the microbiome is a very, very different beast for many other reasons I could suggest as well. And perhaps another one is that just knowing the species that you have in your gut microbiome doesn't tell you the whole story, because you can have multiple strains of one type of species which are incredibly different to each other. So it's more about gene carriage. And so if we look at the microbiome in general, the gut microbiome in general, I would say that if we took all the genetic material from the gut microbiome, we would only really understand what about 30% of the genes are doing. 70% we would call dark matter, from the point of view that we recognize that they're genes, we recognize that they code for proteins, but we have no idea what those proteins do. And so again, it kind of makes it really tricky to kind of translate that to health. And it will be quite a few years before I think we're at the point where we can get a test for microbiome health.